Mesosomes, fact or artifacts. In this particular video lecture, we shall be talking about the mesosome. From the school days, it has been extensively studied about that the mesosome is a bacterial inner membrane fold that help in the respiration process. So as we know in the prokaryotic cell, as there is no mitochondrial structure, so this extended inner membrane structure believed to be participate in the certain cellular mechanism as respiration. So mesosomes or chondrioids, or some scientists call them as a peripheral bodies, are folded invaginations in the inner plasma membrane of the bacteria of the prokaryote that are produced by the chemical fixation. So now still we are not clear about this structure. Only during the chemical fixation these structures are found. The word artifact means that cannot be found in the living structure. So still there is a dilemma whether these are real or false, and used to prepare sample for electron microscopy. So when the scientists were studied this structure and observing under electron microscope, they observed this kind of mesosome, but these were totally absent in the normal living structure. In 1953, first observed by the George Chapman and James Hiller, who referred to them as the peripheral bodies, and they were termed mesosome by the Fitz-James in 1960s. The question is that, is this structure analogous to the cristae in the mitochondria? Because we sometimes equivalent the term bacteria and the mitochondria, because it has been proposed that bacteria is coming to the eukaryotic cell by endosymbiosis. So there are theories that tells during the endosymbiotic relationship, this mitochondria is nothing but a modified bacteria. The answer is yes, as referred by so many scientists. So they are referring as mitochondrial inner folds or their cristae. Similarly, in the bacteria, the inner folds mesosome is analogous structure to the cristae. Mesosome is normally 2 to 4 in number, but that is mostly in gram-positive bacteria. So the presence of mesosome confirms one more criteria, that it is not found in the gram-negative bacteria. So there is a still question, is it an artifact or a fact? So mostly the gram-positive bacteria shows the structure, and more importantly, suppose if you take bacteria A and B, and first one having 4 mesosome and second one having only 1 mesosome, so you can say in the first A bacteria, the respiratory activity and cellular metabolism is higher than the bacterium B. Next, these extensions are told as the form of vesicles, tubules and lamella. Yes, if you see the structure a little bit with care, these are nothing but a fold made up of vesicles, tubule-like structure and lamella. So you can see the vesicles, tubule-like structure and lamella in this extension. So the main contradiction was found when it was fixed. Normally, without any chemicals, when it was fixed without using any chemical, you cannot see any mesosome. But when you use the chemical for fixing, so they observe the mesosome. So that is why so many scientists believe that this is not true, and this is due to the rupturing of this portion of the inner cell membrane, and artifacts are produced during that. And when you fix and frozen, chemical is used and then you frozen it, again the structure reappears. So it is believed that it could be the structure that is not found in the living cells. Recently, new studies has been given that similar fold in the membrane have been observed when exposed to the antibiotics and antibacterial peptide like defensin. And the appearance of this mesosome-like structure may be the result of these chemical, as we have already discussed about it, chemical damaging of the plasma membrane and the cell wall. So if you summarize the entire thing, the structure is mostly present in the gram-positive bacteria and rare in gram-negative. In certain growth conditions are provided, membranes are folded and not the cell wall. It is normally found in the Bacillus bacteria, and 2 to 4 is the general number. It is high in bacteria with the high respiratory activity, and it regulates different processes like photosynthesis as per some belief and research. Vesicle tubules and lamella are the main composition of it. In 1976, we can refer Salton and Owen. They told it is created by the vesicularization of the other half of the lipid bilayer. And if you study the structure of the mesosome, it is very similar to the lipid bilayer, but many proteins are actually absent there. So proteins, like if you say cytochrome C, ATPase, these are not at all present. So lipid component of this membrane is same, but it differs in the protein structure. It's equivalent to the respiratory pockets. So you can say these are respiratory pockets, pocket-like structure for respiration. But since they itself lack outer membrane, so it doesn't have any outer membrane. So you cannot tell this is analogous to the mitochondria, but you can say it is analogous to the cristae. It is devoid of plasma membrane, enzyme like ATPase, dehydrogenase, and cytochrome. According to Laclede in 1973, they unable to transport amino acid, but they may form the septum structure. I can share you some reference if you want to know more about the mesosome. You can study these PDF folders, and in the comment section you please text me what is your idea about the mesosome. It's a fact or structural artifacts achieved during the electron microscopy fixation. Thank you for listening the entire video.